All right, in this second part, I will talk about development of the brain and the physical development of the brain. I mentioned that for the most part, when we're born, we have all the neurons or brain cells that we're going to have, even to when we're adults. The biggest story there is we don't really grow replacement neurons. We really lose them. There are a couple of small exceptions to that, but for the most part, the total number is there. What isn't there at birth are connections. There are some connections being made, but we don't have the complete network of connections between these neurons formed. And by connections, I mean this sort of link between dendrite and axons. So let's look at this neuron drawing for just a second. The dendrites are reaching out from the cell body of the neuron, and they are receiving signals from other neurons. So this could be a cell in the brain, or this could be a cell in the nervous system, maybe bringing a nervous system impulse back from someone's toes up to the brain to be decoded. And so that information comes down from the dendrite to the nucleus, and there it's acted upon or not. And if it's acted upon, an electric impulse is sent down this axon. It's surrounded with a myelin sheath, and we'll talk about that. And when the uh, electric impulse reaches the end of the axon in what's called an axon terminal, it doesn't directly physically connect to the dendrite of the next neuron in that circuit. There's a tiny microscopic gap between that axon and the next dendrite. It's called a synaptic gap. And so that electric impulse causes the release of a chemical called a neurotransmitter. And there are different neurotransmitters that will cross that synaptic gap and link up to the dendrite in the next neuron in that circuit. And so it fits sort of like a lock and a key. And so certain neurotransmitters will fit certain receptors. And when they do, that causes that signal to be received by that next neuron, which in turn passes that information down the dendrite into the main body here of the neuron, where it's either acted upon or not, and that electric impulse is set through the axon to the next neuron. So some things I just said here, some of those terms. Remember, there are synapses, which are those tiny gaps between neurons, and neurotransmitters. So there's a couple of things going on here that can cause physical changes as our brains develop. And one is these connections, the number of these links between the different neurons. So there's the number of those synapses. And the other is the types and proportions of neurotransmitters that are present in the brain. So over the first months of life, there is a really active proliferation of connections being made all through the brain and the nervous system. And the word we use is blooming to describe this rapid growth of connections. And we see this, for example, in an increase in electric activity that we can measure with EEGs at about 18 months to 24 months of age. So this connection, this blooming, is what causes the growth of what's called gray matter, or the mass in the brain. And think about it this way. Our our infant head is large in proportion to our infant body, but it's still not a full-sized human head. And we've already got all the neurons in there that we're going to have. So the growth in the physical size of the brain and then the mass and weight of the brain is going to be due to these, to growth in gray matter and in white matter. And the blooming is that increase in gray matter, these connections being formed. These connections are formed because of stimulation. So a sound comes into the baby's ear and reaches their brain. That's stimulating the temporal lobes, the parts of the brain that are associated with decoding sound. Or a visual image comes in and reaches the occipital lobe in the back, and that's stimulating those parts of the brain. And so those connections are being made. Or movement in space is stimulating those parts of the brain. Or visualizing color or seeing a mother's face. All of those are causing stimuli to reach the brain, which is causing this increased amount of connections. And in fact, we produce too many connections because we overproduce them. And so blooming is followed by what's called pruning, which is eliminating connections we don't need or we're not using. So if a connection isn't used, it will be lost and it won't come back again. And that sounds kind of bad, but it really kind of helps us fine tune our brain to adapt to the environment that we are born into. So think of it this way. This is a metaphor. Imagine you pick up your phone and you call your grandmother's number, and every time you push the button that's her number, her phone rings, and that's what you want it to do. There's a connection between your phone and her phone when you push the right number. But let's say that when every time you push your grandmother's name, her phone rings, but there's also a phone in Manhattan in some office building that rings, and there's also a phone in Arizona out in the desert somewhere in somebody's house that rings. And there's a phone in Moscow in somebody's house that rings, and in Paris and in Tokyo. There's 50 different phones ring every time you call your grandmother's phone, and all these people are answering at the same time. That would be a real mess. That would be pure havoc. So all you want is that connection between your phone and your grandmother's, and you need to get rid of those connections you're not using. So pruning is a good thing for the brain unless our brain is not receiving important stimuli, and then we lose connections that will never get replaced. And this is an example of slides of neurons at birth, and then from 1, 3, 15, and 24 months. Now remember that, and you can see it's easier to look at the one at birth because they're more obvious. See these neurons, or that cell body, or those darker kind of parts, and you can see the axons and dendrites reaching out there. Remember, the number of neurons doesn't change over time, but see how much more density there is at 2 years compared to birth. It's not because there's more neurons there, it's because there are more connections between those axons and dendrites. So there's more circuitry made, there's more uh, communication and linkages between those neurons. And I said that the increase in the mass of the brain is because of gray matter and white matter. So let me also tell you about white matter. When we talked about that first picture of the neuron there, the axon was covered in a myelin sheath. Think about that this way. Here, I'm going to show you something. You can see this. This is the power plug for the laptop I'm talking on. I just unplugged it from the back. Outside that power plug, there's this kind of coating in rubber. And if I cut this open, I would see copper metal uh, lines inside. And that copper is really good at conducting electricity, moving it from place to place. And the rubber is really bad at conducting electricity. It's an insulator. But if I didn't have that rubber there, this plug is plugged in over there. If I was just sitting here holding the copper, it'd be lighting me up right now. But it's not, because the rubber is really bad at conducting the electricity. So that keeps the electricity from dissipating along the way and getting lost. It makes it focus from getting to the outlet over here to my computer. And that's exactly what I want it to do. And that's what myelin does. It's an insulator that goes, remember the axons are sending an electrical impulse. Well, the myelin is insulating that electric transmission. So it wraps around the axons. It's made of fat cells. And this is why it's so important that babies and toddlers and really even preschoolers are getting adequate amount of fat. A young child should never be on a low fat or a non-fat diet. And longer fats like omega-3s are even better at providing this kind of insulin or insulation, not insulin, insulation layer that helps to improve the efficiency of that transmission. So we've got two important parts that are increasing the mass here: the growth of gray matter from blooming and making those connections, and the growth of white matter from myelin, increasing the efficiency of those electrical impulses. And this is just a picture of what that myelination looks like. It's just a sort of a insulator wrapping those cells. So blooming and pruning are shaped by a child's experiences in the world, the stimulation that they're getting from their environment. And there are different timetables for the rate of blooming and pruning in different parts of the brain. That overall in the brain, that overproduction, that rapid connection between those cells reaches its peak at about 4 months old. But different parts of the brain have different timetables. An example there is the prefrontal cortex up here. It peaks at about 1 year of age, and we don't reach our adult density from following pruning until adolescence. So there can be individual differences in timing of blooming and pruning in different parts of the brain. We we know overall tendencies, but heredity and environment can impact timing to some degree. And this is just an example of three different parts of the brain, and there are more parts of the brain, but this is an example of three of the timing of blooming and pruning. So let me tell you about this visual cortex. This is that occipital lobe back here in the back part of the brain. And right here, this line is where birth is. So remember that right here we have the total number of neurons we're just making and losing these connections. So right here in the visual cortex, there's this time in the weeks just following birth where there's not a lot of rapid increase. It's not quite flat, but it's very, very, not very rapid. What's happening is right here, the newborn baby is very nearsighted. They're very limited in their color vision because they don't have as many cones in their eyes. Their point of focus is about 18 inches from their body, which would be about where their caregiver's face is. So there's not a lot of connections being made in the occipital lobe, but then, boom, they really take off. And this corresponds with the physiological development of the muscles, the cones, those kinds of things in the eyes, which are allowing for better focusing. And before 1 year of age, that growth of connections in the visual cortex has stopped, and from there on what we see is pruning. So we're not adding new information. So what would happen here if we were like evil scientists and we wanted to do a terrible experiment, and we put a patch over a newborn baby's eye for the first year of their life? Even though the eye itself physically works fine, if they didn't have any visual information in that first year, they would never be able to see with that eye, because the connections in the brain that are necessary for decoding that visual information would never have formed, and the window is shut. The time will never be there for those to, I mean, there will never be an opportunity for those to be formed after that time. Let's look at this kind of pink line, this purpley line. This is the auditory cortex. This is hearing. And you can see it's really rapid growth right after birth. This is a time when the baby is able to hear phonemes from all different human languages. And at about here, they lose that ability to hear phonemes that aren't spoken around them, that they haven't come into contact with, that they haven't received stimulation from. And there's a little bit of pruning here. They're losing those connections that they're not using. And then it starts to increase again as children's expressive and receptive language skills and vocabulary are growing. And it reaches its peak after age of 3. And from there, we're fine-tuned with pruning. Our orange here is this prefrontal cortex associated with higher ordered thinking and logic and self-control. It grows right after birth, but it doesn't reach its peak until after 3. And from there, it doesn't reach its adult density really until adolescence. So some other aspects of physical development of our brain include lateralization of the cerebral cortex. So the cerebral cortex is that wrinkly outer kind of side of the brain. The wrinkles there give us more surface area in that cortex. And that cortex is made up of different lobes, and it's separated into 2 hemispheres, the right hemisphere and the left hemisphere. As we grow across development, that lateralization, or the amount that each hemisphere is taking on the role of specialized kinds of functioning, increases. So some of the different uh, lobes include the frontal lobe, and we talked about the prefrontal lobe, which is the very front of the frontal lobe. The frontal lobe, which is associated with higher ordered thinking, the occipital lobe, which is vision. I think I can show you better on this picture. The occipital lobe, so this is the front of the brain, this is the back, this is the brain stem. This is what's taking care of sort of the autonomous things like breathing and heart rate. Uh, our cerebellum, which is taking care of things that like balance and coordination that we may not consciously think about. These wrinkly parts, those are the cerebral cortex on the outside layers of the brain. And in the back, we have the occipital lobe, which is taking care of that visual information. We have the parietal lobe, which is things like language and touch. The temporal lobe, which is it's kind of near our ears, and that's where we process hearing, sounds, and it's also associated with learning and feelings. And the frontal lobe, where our higher ordered thinking, memory, and intentional, purposeful movement and behavior where that processing is occurring. And this is looking straight down. So here's the front of the brain and here's the rear. And this is the right hemisphere and this is the left. There's this sort of line going down the middle. This is there's a filament network here called the corpus callosum, where any information that's going from this hemisphere to that or vice versa is passing through that central corpus callosum there. When we're born, our frontal lobe is not mature. We have the ability to regulate our physiological states, and in the first year, we're going to increasingly come under purposeful, intentional control of the innate reflexes that were there when we were born. But our higher ordered cognitive skills are going to emerge over time. Uh, oh, shoot, where did I go? There we go. Um, and I want to really emphasize here the importance of the environment in that blooming and pruning and shaping the brain. So it's an infant's brain development is guided by their experiences in interacting with the environment and with the people around them. So enriched environments enhance the growth of the brain. They enhance the connections that are made. It's almost as if the human brain is born expecting to be stimulated. It's set up ready for the stimulation that we would receive in the environment. And some of those are stimulations that almost any human infant would receive, like hearing the sound of human voices. Others are uh, experiences that may be more closely tied to a particular social group or a particular experience, like hearing a certain sound or a certain phoneme. Uh, we uh, these experiences, visual, tactile, sound, all of them impact different areas of the brain. So as we enter the preschool years, uh, we are seeing increases in size of the brain, elimination of synapses that aren't being used, but we're still seeing some, particularly like in the frontal lobe, uh, up to age 3, still seeing some blooming going on. There's increased lateralization in the preschool years. The right and left brain are increasingly becoming specialized. The brain and the head are showing the fastest growth in the body. By the age of 3, our brain and head is about 75% of our adult size. By age 5, it's about 90% of our adult size, and we're not 90% of our adult height when we enter kindergarten. So that part of the body is growing the fastest. We're seeing increased myelination, and we're also seeing changes in the neurotransmitters, the brain chemistry. For example, the age period of 3 to 6 is a period when we see the neurotransmitter dopamine increase, and that's also associated with self-control. So think about this for a second. There's this little dance between physical development and the chemical development of neurotransmitters. So the prefrontal lobe, which we know is associated with self-regulation, that reaches its full blooming potential at about age 3, followed by age 3 to 6, this increase in dopamine production, which is also governed in self-regulation. Behaviorally, we see that as increased self-control across the preschool years into elementary school. This is just showing that overall growth curve that the brain and head grow faster and then kind of slow down, and overall height and weight is more of a linear progression. Um, Between 3 and 15, some areas will almost double in size through blooming, and then those unneeded cells will be purged. We see different paces of this. So, an important time for the frontal lobe growth is from 3 to 6, but the temporal and parietal lobes, we see continued development all the way through into adolescence. Uh, by the preschool years, by let's say age 3, that frontal cortex, that synaptic density is twice of what it needs to be as an adult or what it will be as an adult. And so that is reduced through pruning, which continues into adolescence. And there are different timetables for the pruning in different parts of the brain, but it's a net loss between preschool and adolescence in the amount of gray matter. And that's just a picture of that prefrontal cortex there where that lobe is. So there are two causes for pruning. There's our genetic heritage and also the specific experiences that we have. And this is really as simple as use it or lose it. Active synapses remain and are strengthened. Inactive ones are eliminated and they're not used. So for example, there are sounds in Arabic that my ear cannot hear. I cannot hear it at all. It's like it doesn't exist to me. And that's because I never heard those sounds during that important time of blooming and pruning when I was an infant. And so I will never have the ability to hear those sounds. Um, on the other hand, the benefit is I can hear segmentation in words more clearly in those languages that I did hear when I was an infant. Um, these maturation can affect our cognitive abilities, especially our higher cognitive functioning, such as memory. The other big change that we see in the preschool years is lateralization. Uh, about one example of this lateralization is that we can observe is handedness. In our society, about 9 out of 10 individuals are right-handed. Of that other 10%, the majority are left-handed, but some are ambidextrous. They show no preference. Let me say something really quick about ambidextrous. Sometimes when people hear ambidextrous, they think sort of super Spider-Man dexterity, and that's not the case. Ambidextrous simply means that you're equally, you don't have a preference between right or left. And what happens there often is uh, children who are ambidextrous show a lag in their fine motor skills because they're not getting as much practice with either the right or the left as a preferred hand. So it's not sort of super dexterity, but it's being able to swap back and forth. Um, Handedness has a genetic influence. We know that in all human cultures, right-handedness is the dominant. Uh, and we know that there's differences in the timing that children show a preference. Some children show a hand preference in utero. So in uh, ooh, we can see already before they're born. I'm trying to think how you'd see hand preference, and it's because they're sucking on the thumbs differently when they're born. We might see in an ultrasound that some babies or some fetus will show a preference with the right or left. But that's a rare number that show it that early. And many preschoolers continue to use both hands and develop a preference later. By the time that they are 6, about 90, I'm going to go back to this because it's not on here. By the time they're 6, about 95% of children have shown a preference for right or left-handed. If they haven't shown that by the time they're 6, that's when we consider that ambidextrous. The majority of right-handed individuals process speech in the left hemisphere of their brain. It's the vast majority. For left-handed individuals, there's more variation. Some use the left side of the brain, some use the right, some it's more diffuse across both hemispheres. And we do see more reading difficulties in left-handed individuals. We also see a higher proportion of mathematicians, musicians, architects, artists, folks who use mathematical and visual thinking among left-handers. That doesn't mean that left-handed people are automatically gifted in math and have difficulty with language. It's just an overall proportion. We also see that if we look at the top scores of tests like the SAT, uh, that top group of scores, about 20% of those who receive them are left-handed, whereas that's only about less than 10% of the overall uh, population. As we reach the elementary school years, the main story of brain development is pruning, as the brain eliminates connections that aren't needs and sort of fine-tunes the neurocircuitry to the demands of the environment. Uh, some new synapses are formed. Some are continued pruned. Development of the frontal cortex is important during this time. Lateralization continues, and so that specialization continues. There's increased development in the corpus callosum, which is allowing for more cohesion and communications from the left to the right side of the brain. And EEG coherence, it's measuring how neurons in different areas of the brain functions with each other. That coherence increases throughout middle childhood. And I'll stop there and talk about sleep in our next slide.